Assalamualaikum warahmatullahi wabarakatuh. Today I will perform an experiment about DNA extraction from banana. Okay, first we have water, then we have soap. Okay, why we use soap? Because during the DNA extraction, detergent will cause the cell to pop open or least so that the DNA is released into solution. Then alcohol add to the solution cause the DNA to precipitate out. Okay, then we have salt. Okay, why we use salt for DNA extraction? Because sodium chloride help to remove proteins that are bound to the DNA. When DNA comes out of solution, it tends to clump together, which make it visible. That's why we use salt. And lastly, we have alcohol. Okay, why we use alcohol? Alcohol is one of essential ingredient of DNA extraction, used for precipitating, storing, and washing DNA. Alcohol with some salt precipitate DNA into a solid visible form. That's why we use alcohol. Okay, let's move the next step. Okay, now let's mix the solution. We have here. First, we must put the into. Okay, we must put the solution into the water. Okay, don't put the water into the solution. Okay, that's the rule of the chemist. Okay, now let's put salt. Okay, I will go a little. So okay, let's stir up. Okay, the mixture and the function of this mixture to make the DNA is visible when we mixing it together. Okay, let's move to the next step. Okay, now let's cut the banana. Okay, we have banana now, and let's put it into the plastic. Okay. Okay, now I have put the banana into the plastic. Okay. Now let's put the solution into the plastic. Alright. Okay, now let's crush the banana to make sure all the cell of banana is mixture with the solution that we have put it. Okay. This process is important. Okay, now we are done. Okay, so let's filter it. Okay, now let's put the alcohol into refrigerator first. Okay, to cool up the alcohol. Okay, because it is important to use cool alcohol because it allows to a larger amount of DNA to be extracted. If the alcohol is too warm, it may cause the DNA to denature or break down. During centrifugation, the DNA condenses into a pellet. When the alcohol is removed, relatively poor DNA will be left behind. Okay, that's why we cool the alcohol first. Okay, now let's put a little alcohol into the solution that we mix with banana. Okay, now we are done. Okay, you can see a little DNA. Okay, you can see a little DNA here. So our experiment is success. Okay, thank you for watching.